Adenosine triphosphate is a nucleoside triphosphate used in cells as a coenzyme. It is often called the molecular unit of currency of intracellular energy transfer. ATP transports chemical energy within cells for metabolism. It is one of the end products of photophosphorylation, cellular respiration, and fermentation and used by enzymes and structural proteins in many cellular processes, including biosynthetic reactions, motility, and cell division. One molecule of ATP contains 3 phosphate groups, and it is produced by a wide variety of enzymes, including ATP synthase, from adenosine diphosphate or adenosine monophosphate and various phosphate group donors. Substrate-level phosphorylation, oxidative phosphorylation in cellular respiration, and photophosphorylation in photosynthesis are 3 major mechanisms of ATP biosynthesis. Metabolic processes that use ATP as an energy source convert it back into its precursors. ATP is therefore continuously recycled in organisms. The human body, which on average contains only 250 grams of ATP, turns over its own body weight equivalent in ATP each day. ATP is used as a substrate in signal transduction pathways by kinases that phosphorylate proteins and lipids, as well as by adenylate cyclase, which uses ATP to produce the second messenger molecule cyclic AMP. The ratio between ATP and AMP is used as a way for a cell to sense how much energy is available and control the metabolic pathways that produce and consume ATP. Apart from its roles in energy metabolism and signaling, ATP is also incorporated into nucleic acids by polymerases in the process of transcription. ATP is the neurotransmitter believed to signal the sense of taste. The structure of this molecule consists of a purine base attached to the 1' carbon atom of a pentose sugar. 3 phosphate groups are attached at the 5' carbon atom of the pentose sugar. It is the addition and removal of these phosphate groups that interconvert ATP, ADP and AMP. When ATP is used in DNA synthesis, the ribose sugar is first converted to deoxyribose by ribonucleotide reductase. ATP was discovered in 1929 by Carl Lochmann, Fisk and Y. Subarow of Harvard Medical School, but its correct structure was not determined until some years later. It was proposed to be the main energy transfer molecule in the cell by Fritz Albert Lippmann in 1941, that is, being the intermediary molecule between energy-yielding and energy-requiring reactions. It was first artificially synthesized by Alexander Todd in 1948. Physical and chemical properties: ATP consists of adenosine composed of an adenine ring and a ribose sugar and 3 phosphate groups. The phosphoryl groups, starting with the group closest to the ribose, are referred to as the alpha, beta, and gamma phosphates. Consequently, it is closely related to the adenosine nucleotide, a monomer of RNA. ATP is highly soluble in water and is quite stable in solutions between pH 6.8 and 7.4, but is rapidly hydrolyzed at extreme pH. Consequently, ATP is best stored as an anhydrous salt. ATP is an unstable molecule in unbuffered water, in which it hydrolyzes to ADP and phosphate. This is because the strength of the bonds between the phosphate groups in ATP is less than the strength of the hydrogen bonds between its products and water. Thus, if ATP and ADP are in chemical equilibrium in water, almost all of the ATP will eventually be converted to ADP. A system that is far from equilibrium contains Gibbs free energy, and is capable of doing work. Living cells maintain the ratio of ATP to ADP at a 0.10 orders of magnitude from equilibrium, with ATP concentrations 5-fold higher than the concentration of ADP. This displacement from equilibrium means that the hydrolysis of ATP in the cell releases a large amount of free energy. 2 phosphorn hydride bonds in an ATP molecule are responsible for the high energy content of this molecule. In the context of biochemical reactions, these anhydride bonds are frequently and sometimes controversially referred to as high-energy bonds. Energy stored in ATP may be released upon hydrolysis of the anhydride bonds. the primary phosphate group on the ATP molecule that is hydrolyzed when energy is needed to drive anabolic reactions is the I-cube phosphate group. Located the farthest from the ribose sugar, it has a higher energy of hydrolysis than either the I+ or minus or I² phosphate. The bonds formed after hydrolysis or the phosphorylation of a residue by ATP are lower in energy than the phosphorn hydride bonds of ATP. During enzyme-catalyzed hydrolysis of ATP or phosphorylation by ATP, the available free energy can be harnessed by a living system to do work. Any unstable system of potentially reactive molecules could potentially serve as a way of storing free energy, if the cell maintain their concentration far from the equilibrium point of the reaction. However, as is the case with most polymeric biomolecules, the breakdown of RNA, DNA, and ATP into simpler monomers is driven by both energy release and entropy increase considerations, in both standard concentrations, and also those concentrations encountered within the cell. The standard amount of energy released from hydrolysis of ATP can be calculated from the changes in energy under non-natural conditions, then correcting to biological concentrations. The net change in heat energy at standard temperature and pressure of the decomposition of ATP into hydrated ADP and hydrated inorganic phosphate is a 20.5 kJ per mole, with a change in free energy of 3.4 kJ per mole. The energy released by cleaving either a phosphate or pyrophosphate unit from ATP at standard state of 1 OMR, ATP plus H, 2O ADP plus pi AI- equals a 30.5 kJ per mole, ATP plus H, 2O AMP plus PPIAI- equals a 45.6 kJ per mole. These values can be used to calculate the change in energy under physiological conditions and the cellular ATP-ADP ratio. However, a more representative value called the energy charge is increasingly being employed. The values given for the Gibbs free energy for this reaction are dependent on a number of factors, including overall ionic strength and the presence of alkaline earth metal ions such as Mg2+ and Ca2+. Under typical cellular conditions, Ig is approximately a 57 kJ per mole. Ionization in biological systems, ATP has multiple groups with different acid dissociation constants. In neutral solution, ATP as ionized exists mostly as ATP4A, with a small proportion of ATP3A. As ATP has several negatively charged groups in neutral solution, it can chelate metals with very high affinity. The binding constant for various metal ions are as Mg2+, Na+, Ca2+, K+, SO2+, and Li+. Due to the strength of these interactions, ATP exists in the cell mostly in a complex with Mg2+. Biosynthesis, the ATP concentration inside the cell is typically 1 euro 10 mm. ATP can be produced by redox reactions using simple and complex sugars or lipids as an energy source. For complex fuels to be synthesized into ATP, they first need to be broken down into smaller, more simple molecules. Carbohydrates are hydrolyzed into simple sugars, such as glucose and fructose. Fats are metabolized to give fatty acids and glycerol. The overall process of oxidizing glucose to carbon dioxide is known as cellular respiration and can produce about 30 molecules of ATP from a single molecule of glucose. ATP can be produced by a number of distinct cellular processes. The three main pathways used to generate energy in eukaryotic organisms are glycolysis and the citric acid cycle oxidative phosphorylation, both components of cellular respiration, and beta oxidation. The majority of this ATP production by a non-photosynthetic aerobic eukaryote takes place in the mitochondria, which can make up nearly 25% of the total volume of a typical cell. Glycolyses. In glycolysis, glucose and glycerol are metabolized to pyruvate via the glycolytic pathway. In most organisms, this process occurs in the cytosol, but, in some protozoa such as the chaetoplastids, this is carried out in a specialized organelle called the glycosome. Glycolysis generates a net 2 molecules of ATP through substrate phosphorylation catalyzed by two enzymes, PGK and pyruvate kinase. 2 molecules of NADH are also produced, which can be oxidized via the electron transport chain and result in the generation of additional ATP by ATP synthase. The pyruvate generated as an end product of glycolysis is a substrate for the Krebs cycle. Glucose. In the mitochondrion, pyruvate is oxidized by the pyruvate dehydrogenase complex to acetyl group, which is fully oxidized to carbon dioxide by the citric acid cycle. Every turn of the citric acid cycle produces 2 molecules of carbon dioxide, 1 molecule of the ATP equivalent guanosine triphosphate through substrate-level phosphorylation catalyzed by succinyl-CoA synthase, 3 molecules of the reduced coenzyme NADH, and 1 molecule of the reduced coenzyme FADH2. Both of these latter molecules are recycled to their oxidized states via the electron transport chain, which generates additional ATP by oxidative phosphorylation. The oxidation of an NADH molecule results in the synthesis of 2 3 ATP molecules, and the oxidation of 1 FADH2 yields between 1 2 ATP molecules. The majority of cellular ATP is generated by this process. Although the citric acid cycle itself does not involve molecular oxygen, it is an obligately aerobic process because O2 is needed to recycle the reduced NADH and FADH2 to their oxidized states. In the absence of oxygen, the citric acid cycle will cease to function due to the lack of available NAD+ and FAD. The generation of ATP by the mitochondrion from cytosolic NADH relies on the malate aspartate shuttle because the inner mitochondrial membrane is impermeable to NADH and NAD+. Instead of transferring the generated NADH, a malate dehydrogenase enzyme converts oxaloacetate to malate, which is translocated to the mitochondrial matrix. Another malate dehydrogenase catalyzed reaction occurs in the opposite direction, producing oxaloacetate and NADH from the newly transported malate and the mitochondrion's interior store of NAD+. A transaminase converts the oxaloacetate to aspartate for transport back across the membrane and into the intermembrane space. In oxidative phosphorylation, the passage of electrons from NADH and FADH2 through the electron transport chain powers the pumping of protons out of the mitochondrial matrix and into the intermembrane space. This creates a proton motive force that is the net effect of a pH gradient and an electric potential gradient across the inner mitochondrial membrane. Flow of protons down this potential gradient that is, from the intermembrane space to the matrix provides the driving force for ATP synthesis by ATP synthase. This enzyme contains a rotor subunit that physically rotates relative to the static portions of the protein during ATP synthesis. Most of the ATP synthesized in the mitochondria will be used for cellular processes in the cytosol. Thus it must be exported from its site of synthesis in the mitochondrial matrix. The inner membrane contains an antiporter, the ADP-ATP translocase, which is an integral membrane protein used to exchange newly synthesized ATP in the matrix for ADP in the intermembrane space. This translocase is driven by the membrane potential, as it results in the movement of about 4 negative charges out of the mitochondrial membrane in exchange for 3 negative charges moved inside. However, it is also necessary to transport phosphate into the mitochondrion. The phosphate carrier moves a proton in with each phosphate, partially dissipating the proton gradient. Beta oxidation. Fatty acids can also be broken down to acetyl-CoA by beta oxidation. Each round of this cycle reduces the length of the acyl chain by 2 carbon atoms and produces 1 NADH and 1 FADH2 molecule, which are used to generate ATP by oxidative phosphorylation. Because NADH and FADH2 are energy-rich molecules, dozens of ATP molecules can be generated by the beta oxidation of a single long acyl chain. The high energy yield of this process and the compact storage of fat explain why it is the most dense source of dietary calories. Fermentation. Fermentation entails the generation of energy via the process of substrate-level phosphorylation in the absence of a respiratory electron transport chain. In most eukaryotes, glucose is used as both an energy store and an electron donor. The equation for the oxidation of glucose to lactic acid is C. 6 hours, 12O, 62CH, 3CH, O, COOH plus 2 ATP, anaerobic respiration. Anaerobic respiration is the process of respiration using an electron acceptor other than O2. In prokaryotes, multiple electron acceptors can be used in anaerobic respiration. These include nitrate, sulfate or carbon dioxide. These processes lead to the ecologically important processes of denitrification, sulfate reduction and acetogenesis, respectively. ATP replenishment by nucleoside diphosphate kinases. ATP can also be synthesized through several so-called replenishment reactions catalyzed by the enzyme families of nucleoside diphosphate kinases, which use other nucleoside triphosphates as a high-energy phosphate donor, and the ATP, guanidophosphotransferase family. ATP production during photosynthesis. In plants, ATP is synthesized in thylakoid membrane of the chloroplast during the light-dependent reactions of photosynthesis in a process called photophosphorylation. Here, light energy is used to pump protons across the chloroplast membrane. This produces a proton motive force and this drives the ATP synthase, exactly as in oxidative phosphorylation. Some of the ATP produced in the chloroplasts is consumed in the Calvin cycle, which produces triose sugars. ATP recycling. The total quantity of ATP in the human body is about 0.2 mol. The majority of ATP is not usually synthesized de novo, but is generated from ADP by the aforementioned processes. Thus, at any given time, the total amount of ATP plus ADP remains fairly constant. The energy used by human cells requires the hydrolysis of 100 to 150 moles of ATP daily, which is around 50 to 75 kg. A human will typically use up his or her body weight of ATP over the course of the day. This means that each ATP molecule is recycled 500 to 750 times during a single day. ATP cannot be stored, hence its consumption closely follows its synthesis. However, a total of around 5 g of ATP is used by cell processes at any time in the body. Regulation of biosynthesis. ATP production in an aerobic eukaryotic cell is tightly regulated by allostric mechanisms, by feedback effects, and by the substrate concentration dependence of individual enzymes within the glycolysis and oxidative phosphorylation pathways. Key control points occur in enzymatic reactions that are so energetically favorable that they are effectively irreversible under physiological conditions. In glycolysis, hexokinase is directly inhibited by its product, glucose 6-phosphate, and pyruvate kinase is inhibited by ATP itself. The main control point for the glycolytic pathway is phosphofructokinase, which is allostrically inhibited by high concentrations of ATP and activated by high concentrations of AMP. The inhibition of PFK by ATP is unusual, since ATP is also a substrate in the reaction catalyzed by PFK. The biologically active form of the enzyme is a tetramer that exists in two possible conformations, only one of which binds the second substrate fructose 6-phosphate. The protein has two binding sites for ACT. The active site is accessible in either protein conformation, but ATP binding to the inhibitor site stabilizes the conformation that binds F6P poorly. A number of other small molecules can compensate for the ATP-induced shift in equilibrium conformation and reactivate PFK, including cyclic AMP, ammonium ions, inorganic phosphate, and fructose 1, 6 and 2, 6-biphosphate. The citric acid cycle is regulated mainly by the availability of key substrates, particularly the ratio of NAD+ to NADH and the concentrations of calcium, inorganic phosphate, ATP, ADP, and AMP. Citrate A. The molecule that gives its name to the silea A. is a feedback inhibitor of citrate synthase and also inhibits PFK, providing a direct link between the regulation of the citric acid cycle and glycolyses. In oxidative phosphorylation, the key control point is the reaction catalyzed by cytochrome C oxidase, which is regulated by the availability of its substrate A. The reduced form of cytochrome C. The amount of reduced cytochrome C available is directly related to the amounts of other substrates. which directly implies this equation. Thus, a high ratio of NADH to NAD+ or a low ratio of ADP, pi to ATP imply a high amount of reduced cytochrome C and a high level of cytochrome C oxidase activity. An additional level of regulation is introduced by the transport rates of ATP and NADH between the mitochondrial matrix and the cytoplasm. Functions in cells, metabolism, synthesis, and active transport. ATP is consumed in the cell by energy-requiring processes and can be generated by energy-releasing processes. In this way ATP transfers energy between spatially separate metabolic reactions. ATP is the main energy source for the majority of cellular functions. This includes the synthesis of macromolecules, including DNA and RNA, and proteins. ATP also plays a critical role in the transport of macromolecules across cell membranes, for example exocytosis and endocytosis. Roles in cell structure and locomotion. ATP is critically involved in maintaining cell structure by facilitating assembly and disassembly of elements of the cytoskeleton. In a related process, ATP is required for the shortening of actin and myosin filament cross bridges required for muscle contraction. This latter process is one of the main energy requirements of animals and is essential for locomotion and respiration. Cell signaling, extracellular signaling. ATP is also a signaling molecule. ATP, ADP, or adenosine are recognized by purinergic receptors. Purinoreceptors might be the most abundant receptors in mammalian tissues. In humans, this signaling role is important in both the central and peripheral nervous system. Activity-dependent release of ATP from synapses, axons and glia activates purinergic membrane receptors known as P2. The P2Y receptors are metabotropic, that is G-protein coupled, and modulate mainly intracellular calcium and sometimes cyclic AMP levels. Though named between P2Y1 and P2Y15, only 9 members of the P2Y family have been cloned, and some are only related through weak homology and several do not function as receptors that raise cytosolic calcium. The P2X ionotropic receptor subgroup comprises 7 members, which are ligand-gated Ca2+- permeable ion channels that open when bound to an extracellular purine nucleotide. In contrast to P2 receptors, purinergic nucleoside triphosphates like ATP are not strong agonists of P1 receptors, which are strongly activated by adenosine and other nucleosides. P1 receptors have A1, A2A, A2B, and A3 subtypes, all of which are G-protein coupled receptors, A1 and A3 being coupled to GI, and A2A and A2B being coupled to GS. All adenosine receptors were shown to activate at least one subfamily of mitogen-activated protein kinases. The actions of adenosine are often antagonistic or synergistic to the actions of ATP. In the CNS, adenosine has multiple functions, such as modulation of neural development, neuron and glial signaling and the control of innate and adaptive immune systems. Intracellular signaling. ATP is critical in signal transduction processes. It is used by kinases as the source of phosphate groups in their phosphate transfer reactions. Kinase activity on substrates such as proteins or membrane lipids are a common form of signal transduction. Phosphorylation of a protein by a kinase can activate this cascade, such as the mitogen-activated protein kinase cascade. ATP is also used by adenylate cyclase and is transformed to the second messenger molecule cyclic AMP, which is involved in triggering calcium signals by the release of calcium from intracellular stores. This form of signal transduction is particularly important in brain function, although it is involved in the regulation of a multitude of other cellular processes. DNA and RNA synthesis. In all known organisms, the deoxyribonucleotides that make up DNA are synthesized by the action of ribonucleotide reductase enzymes on their corresponding ribonucleotides. These enzymes reduce the sugar residue from ribose to deoxyribose by removing oxygen from the 2' hydroxyl group. The substrates are ribonucleoside diphosphates and the products deoxyribonucleoside diphosphates. All ribonucleotide reductase enzymes use a common sulfhydryl radical mechanism reliant on reactive cysteine residues that oxidize to form disulfide bonds in the course of the reaction. RNR enzymes are recycled by reaction with thiodoxin or glutadoxin. The regulation of RNR and related enzymes maintains a balance of dNTPs relative to each other and relative to NTPs in the cell. Very low dNTP concentration inhibits DNA synthesis and DNA repair and is lethal to the cell, while an abnormal ratio of dNTPs is mutagenic due to the increased likelihood of the DNA polymerase incorporating the wrong dNTP during DNA synthesis. Regulation of or differential specificity of RNR has been proposed as a mechanism for alterations in the relative sizes of intracellular dNTP pools under cellular stress such as hypoxia. In the synthesis of the nucleic acid RNA, adenosine derived from ATP is one of the 4 nucleotides incorporated directly into RNA molecules by RNA polymerases. The energy driving this polymerization comes from cleaving off a pyrophosphate. The process is similar in DNA biosynthesis, except that ATP is reduced to the deoxyribonucleotide DATP, before incorporation into DNA. Amino acid activation in protein synthesis. Aminosyl tRNA synthase enzymes utilize ATP as an energy source to attach a tRNA molecule to its specific amino acid, forming an aminosyl tRNA complex, ready for translation at ribosomes. The energy is made available by ATP hydrolysis to adenosine monophosphate as 2 phosphate groups are removed. Binding to proteins. Some proteins that bind ATP do so in a characteristic protein fold known as the Rossmann fold, which is a general nucleotide binding structural domain that can also bind the coenzyme NAD. The most common ATP binding proteins, known as kinases, share a small number of common folds. The protein kinases, the largest kinase superfamily, all share common structural features specialized for ATP binding and phosphate transfer. ATP in complexes with proteins, in general, requires the presence of a divalent cation, almost always magnesium, which binds to the ATP phosphate groups. The presence of magnesium greatly decreases the dissociation constant of ATP from its protein binding partner without affecting the ability of the enzyme to catalyze its reaction once the ATP has bound. The presence of magnesium ions can serve as a mechanism for kinase regulation. ATP analogues. Biochemistry laboratories often use in vitro studies to explore ATP-dependent molecular processes. Enzyme inhibitors of ATP-dependent enzymes such as kinases are needed to examine the binding sites and transition states involved in ATP-dependent reactions. ATP analogues are also used in X-ray crystallography to determine a protein structure in complex with ATP, often together with other substrates. Most useful ATP analogues cannot be hydrolyzed as ATP would be. Instead they trap the enzyme in a structure closely related to the ATP-bound state. Adenosine 5-gamma-triphosphate, is an extremely common ATP analog in which one of the gamma-phosphate oxygens is replaced by a sulfur atom. This molecule is hydrolyzed at a dramatically slower rate than ATP itself and functions as an inhibitor of ATP-dependent processes. In crystallographic studies, hydrolysis transition states are modeled by the bound vanadate ion. However, caution is warranted in interpreting the results of experiments using ATP analogues, since some enzymes can hydrolyze them at appreciable rates at high concentration. See also, adenosine diphosphate, adenosine monophosphate, adenosine tetraphosphatase, ATPases, ATP test, ATP hydrolysis, citric acid cycle, cyclic adenosine monophosphate, nucleotide exchange factor, phosphagon, photophosphorylation. References. External links: ATP bound to proteins in the PDB, Science Aid, Energy ATP and Exercise, PubChem entry for adenosine triphosphate, KEGG entry for adenosine triphosphate.